So we finished the structure of DNA, how it's structured, how it's packaged in the cell, and how it replicates. These people over here, bulbs out, we're working on it. All right? But now the next step is that DNA has all the code, right? It has all the genetic information. Scientists have proven through structure and through several other experiments and research that DNA is the genetic material and it passes on genes. Okay? So DNA includes the code for all the genes, but DNA stays where in a eukaryotic cell? It stays in the nucleus. Therefore, does DNA do any of the functions that the cell needs to do? No. All right. Those functions are carried out by what? Proteins. Okay? So DNA has the code for all the proteins that do all the functions. Okay? So now the idea is, how do we get from DNA with a gene code to the protein that's going to do the work? And we know that RNA, ribonucleic acid, is the bridge between DNA and protein. All right? So this next chapter is really getting from where the code is, DNA, transcribing the RNA, right? And then translating the overall code into a functional protein. Okay? So we're going to go through transcription and translation and eventually protein synthesis. Okay? It's not there. Okay? No. The biotech stuff?
We don't need to know that?
No. Sorry. Okay? We went over that the other day. So, RNA is structured similar to DNA. It is composed of ribonucleotides. It's composed of nucleotides, right? Yes? It has a 5-carbon sugar. Okay? That strand of RNA is composed of a phosphodiester backbone. All right? The difference is, RNA is what? What is different?
It's one-stranded.
Single-stranded. What? Emily? There's a ribose for a sugar, okay, instead of a deoxyribose. What else is different in RNA from DNA? Doesn't have the something. What doesn't it have?
Thymine? No.
It doesn't have thymine. It has what instead? Uracil. Single-stranded, right? Uracil instead of thymine, ribose as the 5-carbon sugar. Okay? Those are the 3 basic differences between RNA and DNA. Similarities are it's composed of a phosphodiester backbone, there's a sugar, there are nitrogenous bases. Okay? So, but getting from DNA to protein requires that intermediate RNA, and so that's transcription. Okay? Transcription is the first process we're going to talk about. That's the process of taking the code that's found in DNA and transcribing it into an RNA. All right? So transcription. And then translation is deciphering the code that has been transcribed into the mRNA. All right? And that's done by ribosomes. All right? So we're going to talk about messenger RNA. That's the exact code off of DNA. All right, that's produced. And then you have that ribosomes will decode that mRNA to produce a chain of amino acids. Okay? Now, some inherent differences between prokaryotes and eukaryotes. Okay? What is the main difference between a prokaryote and a eukaryote? Well, a eukaryotic cell and a prokaryotic cell is still unicellular. Organelles, specifically which one? A membrane-defined nucleus, okay, which is going to separate where the DNA is found in a eukaryotic cell and where it's not found in a eukaryotic cell. Okay? Really, that's really the bulk of what a nuclear membrane does, what the nucleus is. It contains the DNA. All right? So in prokaryotes, there is no containment. All right? So in prokaryotes, transcription can start and translation can start because there's no fence. There's no barrier between where the ribosomes are in the cytoplasm, in the cytosol, and where transcription's happening in where the DNA is. Okay? So in prokaryotes, because there's no membrane-bound nucleus, transcription and translation start at the same time. Okay? But because of the separation in eukaryotic cells, there's a breakage. The mRNA is produced first and then travels out of the nucleus to where the ribosomes are. And then translation starts. So there's a break in the pattern, in the sequence. All right? And the transport of mRNA out of the nucleus is an important step. Okay? It is a step, provides a step of regulation. If the cell no longer wants that gene to be expressed, it can prevent the mRNA from leaving the nucleus for whatever reason, and then it won't be translated into protein. All right, as an example, towards the end of the chapter. Okay? Here's your overall schematic of what's happening. Transcription, along with replication in eukaryotic cells, happens in the nucleus. All right? The mRNA is transcribed. It is then processed. We'll go through the steps of processing. All right? But it's processed. It's basically processing is preparation for sending it out of the nucleus. Okay? Questions on that? And then outside of the nucleus, you get translation. In a prokaryotic cell up here on your left, there's no separation, so transcription starts and mRNA begins, ribosome binds, translation begins. Okay, so everything happens simultaneously. Okay, it does show there's a little arrow, but it does happen simultaneously. Okay, so a little vocabulary. We're going to talk about what a primary transcript is. Okay, that is the initial RNA that's produced off of a gene. Okay, it is not edited. It is not processed. A primary transcript is not ready to exit the nucleus. Okay? It's also called a pre-mRNA. All right? Primary transcript, pre-mRNA are synonymous. Okay? Now, this next part right here, okay, the central dogma, is the concept that cells are governed by a cellular chain of command, which is DNA codes for RNA and codes for a protein. As recently as 2 weeks ago, there was a paper published that challenged this position, where a protein directed the production of another protein. Okay? So this has long been held as the central dogma of molecular biology, that genetic information flows from the DNA to RNA to protein. All right? But most recently, and I'm going to have you guys read post an article, you're going to read a little explanation about it, that this is now being challenged by current research to show that this is not just a one-way street anymore. Okay? There's much we know about how proteins function that may be different. Okay? But for the longest time, this has been we call it the dogma because it is a pillar belief in biology. DNA to RNA to protein. And that is no longer the headlines where we have to change the textbooks. Okay? For now, we're going to go through what's most known about it. You will read an explanation about what that is, about the other challenge to this. But understanding how DNA, the RNA, the protein works is important, and first important, and then how is it going to be different, okay? So, the genetic code. The code itself is what we call a triplet code, okay? There are at least 3 base pairs that are necessary to code for an amino acid, which will then be part of your proteins, okay? So, the instructions for assembling amino acids are found along the letters of the RNA that we're talking about, that is transcribed off the DNA that codes for the gene. The RNA is going to transcribe that code, and that code is in 3 base pair words, okay? And then each of those 3 base pairs codes for a specific amino acid, which then determines the sequence of proteins, all right? The primary sequence. So how many nucleotides correspond to an amino acid? How many? 3. All right, so there's your triplet code. All right, the words of the gene are transcribed into complementary, non-overlapping 3 nucleotide chunks of RNA, words of RNA. All right, so ATG, that's 1. GTC, that's another. CAA, that's another. Well, what did I say wrong there? T. Good. All right, so that would AUG would be RNA, correct? All right, GUA would be RNA. And then those so AUG codes for methionine. That is an example of decoding and translating the triplet into an amino acid. All right, there are 20 amino acids. All right, based upon the 3-letter code and having 4 options for each letter. It's like 4 to the 3rd power. That means there are 64 possible codons, possible triplet combinations. Okay, I saw a puzzle look back there, so if we go like this. Okay, so if I have, here's 1, 2, 3. And each of these 3 spaces can be A, U, C, G, right? Each of them can be one of those. So to figure out how many combinations that is, you take the 4 possibilities to the 3rd power, that equals 64 different combinations for a triplet. Okay? How many amino acids do we have? 20. How many possible codes do we have for all those amino acids? 64. What does that tell you? They either not all used or some of them are reused. They're used more than once. And that is what we call the redundancy of the code. Okay? Redundancy meaning that in some cases there are 4 options for one amino acid. Okay? In other cases, there's only 1. All right? So depending upon the amino acid you're talking about, in some cases, this code is redundant. Right? Meaning that it is those codons, those triplets, are reused. Okay? So here's our DNA template, where RNA is what to DNA? How is RNA produced? If DNA forms the template, RNA is going to be produced based upon what rules? The base pairing rules, right? A to U, C to G, G to C. If there's a T in DNA, it's going to be what in RNA? If there's a complementarity. If there's a T in DNA, it's going to be what in RNA? A. So let's look. Let's take which one of the strands here in DNA, top or bottom, is the template strand for that RNA. And I'm standing right in the middle of where the people are looking. So, is it the top or the bottom strand? Top, right? We'll do it this way. A to U, C to G, C to G. Right? You could also know that The non-template strand in DNA, if you took out all the T's and replaced it with U's, it would be equal to what you have in the RNA. Okay? The non-coding strand, right? If you take out this T, these T's, and replace them with U, they would be equal to what you have in the RNA. Okay? Single-stranded RNA. And now we're going to read that in 3 base pair chunks for translation, and you're going to get your tryptophan, your phenylalanine, glycine, serine in 3 base pair chunks. Okay? That's the triplet code. That's the basic overview of transcription and translation. Okay? Okay, so we went through the template strand, all right? One little thing to note, the template strand is always the same strand for any given gene. Why? If we went back and I said, all right, here's our template strand, say this one's our template strand, and this is A, C, C, T, A, G, G, C. 1, 2, 3, 1, 2, 3, A. Okay? What's the non-template strand going to be? What's complementary? What's the other strand of DNA going to be? T. Okay. Now, what's the mRNA going to be? Oop. U, G, G, A, U, C, C, G, U. What would the mRNA be if I used the non-template strand as a template strand? So we're going to use this one instead of the top one. Is it going to give you the same RNA? No. Okay? So for any given gene, the template strand needs to be the same every time, or else you're not going to get the same RNA. Right? Not the same RNA leads to a different protein. Therefore, it's a different gene. Right? Different function. All right, so during translation, the codons are read in the 5' to 3' direction. Each chunk of 3, each codon represents 1 amino acid. All right, so each codon represents 1 amino acid. AUC is glycine. It's not phenylalanine. Okay? Now, 1 amino acid could have more than 1 codon. Right? AUG could be glycine. AUC could be glycine. AUU could be glycine. But AUU does not code for more than 1 amino acid. Okay? 1 codon, 1 amino acid. Right? So the 64 codons were deciphered in there we go. Were deciphered in the 1960s. Of the 64, right, 61 code for amino acids. So some of them do not. If it does not code for an amino acid, what do you think it codes for? A stop codon. All right? Basically, it doesn't mean, it's not an inherent stop signal. And we're going to go through the particulars of this, specifics of it. But an amino acid can't be brought in because it doesn't fit with any other, the messenger or the tRNAs. So no amino acid can be brought in, so the ribosome ends up separating and stopping translation. Okay? So we call it a stop codon, but it's not like a cutoff. All right? We'll see it. We talk about the redundancy, and it's not ambiguous. No codon specifies more than 1 amino acid. All right? So redundant, but not. Now, those codons need to be read in what we call the right reading frame. Okay? If I have an RNA, which is U, G, G, oh, that's an awful G, 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 C, G, A, U, U, U. Okay? That's 3, that's 3, that's 3. That means, for hypothetically speaking, this could be valine, alanine, and glutamic acid, as an example. That's not exactly what it is, but whatever. Point being, those 3 codons code for those 3 amino acids. Okay? If read in the right reading frame. if I shift it over one base pair of the codon. So instead of UGG, I'm going to have what? Let's shift it over one. Start at the G instead of the U. GGC. Is that going to code for valine? No. Or it may or may not. But more likely than not, it's not. More likely it's not going to code for the same amino acid. All right? So in order to have the right gene or the right protein from the right gene, has to be in what we call the right reading frame. Okay? And we're going to go through reading frames as we get through the specifics of this as well. Okay? Codons are read one at a time in a non-overlapping fashion based upon the structure of a ribosome. So a ribosome is going to bind like this, and it's going to position the mRNA so that it is in the right position and will read 3 base pair chunks. Okay? So the structure of the ribosome and the structure of the mRNA is important in the function of producing the amino acid. Okay? Here are your codons. Okay? You don't need to know all this if I ask you to do it, and I will provide this sheet for you. But it's important that you understand what you're looking at. All right? On the left-hand side is your first base. On the left-hand, along the top is your second base, and along the right-hand side is your third base of the triplet code. Okay? So you go inside here, you find your CCC is proline. Well, so is CCU, CCA, and CCG. Okay. But AUG is the only one that codes for methionine, which is usually your start codon. Okay? And you could go through all the others. Here are your 3 stops, UAA, UAG, and UGA. Of course, the UGA is the stop codon. Figures. Okay? Questions on the codons themselves. All right, the basics of it. Yeah.
Why is there 1 start but 3 stops?
For the structure of mRNA, in order for it to get out of the nucleus, that first amino acid, that first codon, needs to be a methionine. Okay? So the codon is AUG, right? There's that adenine is methylated and provides what we call a G-methyl cap on that adenine, which you can't put on other codons or on other adenines. The way genes are typically structured, they begin with the CAT, right? As a transcription start site is a CAT. So that's also part of the genetic code. The 3 stops are just nonsense. They don't code for an amino acid, so that is the only reason why. All right, evolution. Genetic code is nearly universal, shared by the simplest bacteria and the most, over here.
If you're counting all the amino acids in a polypeptide, would you still count the one that codes for a stop, or would you?
If you're going to count the amino acids, no. If I count the codons, yes. So if I gave you a length of RNA and asked how many codons are present, you would count that as a stop. If I asked you how many amino acids does this length of RNA code for, it would be, if it was 15 base pairs in RNA, that would be 5 codons, but only 4 amino acids. Okay, because it does not technically code for an amino acid. Okay? Evolution. Now, this fact that the code, the fact that codons are held and conserved between species is an awesome benefit to research and medicine. All right? Because we can take genes out of humans, put them in bacteria, and the bacteria are going to produce them just like humans were producing them. This is how we produce insulin for insulin-dependent diabetics. Take the human gene, all right? You molecular biology-wise, you use some techniques and amplify it, put it into bacteria, and grow up these huge these huge reactors about the size of this room, full of bacteria growing. Okay? And all of that bacteria have that gene inside it, and then they're producing that protein. Then we collect all that bacteria, isolate the protein, and we can put it and market it as a insulin, a human insulin form. Okay? It allows translation and production of proteins that are not necessarily unique or inherent to humans. Humans do not phosphoresce. They do not express bioluminescence, do they? Okay? Neither do pigs. But neither do plants, for the most part. Okay? Jellyfish do. Fireflies do. Right? Fireflies, the fact that you see them, in summer, flying around the grass, whatever. When they light up green, that is an example of bioluminescence. We took that firefly gene and we can put it into a pig. Or in this case, this is a jellyfish gene, same thing. Okay? And you can make a pig glow yellow. They have dogs that glow red. They have cats that glow green. They have cows that glow green. And on and on and on. All because they are producing a bacterial or a jellyfish or whatever gene that's not usually inside them, but because the cell has that machinery and the code is conserved, can be done. All right? And we use this in research to show, where things are in the cell, what's happening to this to this pig, etc. in biology. Questions on that? Question on the overall process and what it does. So we're going to go from DNA, we're going to transcribe the code from DNA to RNA, and then translate it from RNA to DNA, or RNA to protein. All right? Transcription is that first step of gene expression. So to get a gene expressed in the cell, we have to transcribe the code and then translate it to protein. Okay? So how is the overall process of transcription? What do we have to make in transcribing something? What are we going to make? What's the product? What are we going to produce in transcription? RNA. Thank you. Someone's listening. All right. So RNA. What are we going to use to produce that RNA? DNA as a template. Okay? So in order for this to happen, what has to happen to the DNA? The DNA template is usually found how? In a double helix, right? Bound to one another, double-stranded. So we're going to use that DNA as a template to produce a RNA. What has to happen? They have to split them apart, right? So we're going to split them apart, and then an enzyme, RNA polymerase, is going to recognize that open DNA and is going to produce the new RNA strand. Right? RNA nucleotides are assembled in a 5' to 3' direction. Why? Why in a 5' to 3' direction? The free 3' OH. Okay? So we're always going to add it. Okay? But unique about RNA polymerases, they don't require a primer. Okay? It's not like DNA polymerase where they has to have a 3. They can incorporate the RNA nucleotides on their own. Okay? So no primer required. 5' to 3' direction, incorporating on its own. Okay? But where does it start from? Okay? How does a cell direct where an RNA polymerase is going to go? Because there's always going to be areas of the DNA that are open. Okay? But we want it to specifically go to certain parts. Okay? And genes are structured so that the part where that's not coded for the mRNA helps direct how that part that is coded is expressed. Okay? So what you see up here is two parts of a DNA, a transcriptional unit. All right? This right here is a full part of DNA. Okay? And the dark blue part represents the gene. Okay? That's going to code for a protein at some point. All right? The light blue part up here, so at the bottom end, or the beginning and the end part, are what we call the untranslated regions. Why are they untranslated? Well, this part is the untranslated region. Because it's not going to code for the protein, all right? It may be included in the RNA, but it's not going to code for the protein, okay? And then over here in this light blue part, this is the promoter. The promoter region is going to direct how the gene that you're looking at is transcribed, okay? You'll notice this is RNA polymerase. Where does RNA polymerase bind to? Does it bind to the promoter or to the gene? What do you think? Is it important that the RNA polymerase bind where? Should it bind at the promoter or on the gene itself? If it bound to the gene, it would look something over here. Okay? Then what's going to happen? Say if this is that's where transcription is going to start at the end of that. Okay? The promoter directs where transcription is going to begin by binding RNA polymerase in the right spot. Okay? Promoters of DNA, and we'll talk about this next chapter, have a certain structure that will bind and place RNA polymerase in the right spot. They have things called what we call TATA boxes. TATA, was that found in RNA or DNA? DNA. Right? This TATA box is usually found in the promoter region, and it actually helps bind RNA polymerase. It is at a conserved spot upstream or before a gene is going to be transcribed, a spot or a transcription start site. Okay? So it's usually about 10 to 35 base pairs upstream of the transcription start site. Okay? So that allows this to open and then begin processing, right, polymerizing an RNA transcript. Okay? It adds nucleotides in the 5' to 3' direction, producing the transcription of the transcript until it reaches a termination signal. Okay? So a termination signal is inherent of the DNA. All right? It actually, the RNA polymerase loses ability to bind and separates, and that's your termination signal. Pretty simple. What you end up with is a complete or a completed RNA transcript. All right? Okay, your promoter and terminator. Okay, the promoter, you have a terminator also, which is part of the transcription, part of the DNA. It's a sequence of DNA that determines that when that gene ends. All right? So then the stretch of DNA that's transcribed is called your transcriptional unit. All right? Some of that transcriptional unit will not become protein. That's what we call the untranslated region. All right, so 3 parts of transcription: initiation, elongation, and termination. Initiation involves what? What do we have to do? Unwind the double helix, right, and bind the RNA polymerase in the right spot. Elongation is going to be what? Adding nucleotides. Adding ribonucleotides as it moves down the gene. And then termination is stopping adding nucleotides and separating RNA polymerase from the DNA and pushing out the RNA transcript. RNA polymerase. How does it get there? All right? I said it's part of the sequence of DNA, usually a TATA box, but there are other factors that also bring it in. A lot of transcription factors, which we'll go through in chapter 14, will help direct when and where RNA polymerase is placed in order for it to bind to the transcriptional unit and produce RNA. All right? So they mediate the binding of RNA polymerase and the. Okay? Really what you end up with is a transcription initiation complex. Bunch of transcription factors, whatever they might be, ushering RNA polymerase to the right spot, finding the ta-ta box, and beginning at the right starting point. All right? So their ta-ta box is crucial in forming the initiation complex. Okay? So that's your transcription. All right? We're going to produce our pre-mRNA and then go on. Okay? There's your ta-ta box, transcription factors, and so on. Questions on the overall process of transcription, translation, and gene expression, and the process of the specific process of transcription, all right, the specifics of it, how the RNA polymerase is brought in, etc. No? Yes? On Monday, snow willing, we will go through translation. All right? I'm going to try to pick up the pace here because we've lost a couple classes and we're already behind. So expect I'll be picking up the pace a little bit.